Okay, welcome to the Imperial Festival. I hope you had already a chance to enjoy all the attractions that Imperial is offering, all the research we're doing here, all the exciting work. And this lecture that I'm going to give here is about the work we do in my research group and what we find interesting. And what we find interesting has something to do with neurotechnology. And neurotechnology has to do with neurology or neuroscience and technology. And we are bringing these two things together. That's why I'm a neuroscientist, but I work in the bioengineering department, in the computing department. My lab works across these disciplines, and I'm going to talk to you about brains behaving badly. So, before we start, we should maybe establish that all our brains can behave badly, and I'm going to illustrate that with a simple video here. So, observe this speaker, and I would like to do the following experiment. I would like to ask everyone who's sitting to the left of the center to close their eyes, and everyone to the right to keep their eyes open. And what I want you to do is just to listen what this gentleman has to say. And one more time. Okay. Now, clearly we've heard all the same thing. So may I ask somebody on the right-hand side, what have you heard? A volunteer. Da. 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 And anyone on the left-hand side, what have you heard? Baba. Are you sure you're sitting in the same room? Okay. So I'll give you a chance. Everyone on the left-hand side, please do the opposite of what you did before. And on the right-hand side, you can keep your eyes closed this time. Okay? Ready? I'll do it one more time. So it seems very clearly that you're hearing with your eyes and not with your ears, because it depends upon whether you open or close your eyes what you hear. And in fact, what's happening here is the gentleman in the video is saying gaga. And we simply play back him once saying baba and once dada. The problem is, or the challenge is, that when you look at people speaking, your brain needs to make sense out of all the information that arrives. And that making sense of the information that arrives and interpreting the world, that's what we call perception. So perception has nothing to do with the real data, with the photograph you take with the camera. It's what your brain puts together to make out and define what reality is. And here we can basically trick your brain, make your brain behave badly by giving the illusion that the sound coming out of the mouth is a different one by making the mouth move differently. And so this is a very active process of perception, and I'm going to show you during my talk more examples of that. So I spoke that we're engineers and neuroscientists at the same time. Now, how do the two things come together? And that's best I can illustrate it with another visual illusion. So I would like to ask those in the first rows to identify whether this is a man or a woman. Man. And those in the last rows, it's a woman. Okay. So you clearly see a pattern coming up here. Your brains are behaving badly. So let me simply show you the same picture, and I'll make the picture a lot bigger. So now all of you should see a man. Yeah? And when I make this picture a lot smaller, all of you should see a woman. Okay? So you can be Marilyn Monroe and Albert Einstein at the same time. So how do we do that? Well, actually, if you look at this large picture, you may see the trick. So the features of Einstein are like this fine hair, the sharp shapes of his eyes, his frizzy hair up here, the tie, and so forth. They change very rapidly. It goes from black to white to black very quickly. While the features of Marilyn Monroe are much smoother. They go from black to something like gray to white, and then slowly back to gray and white. And this basically means that your brain does something engineers call Fourier decomposition. it tries to decompose the image into images of different scales of change. And that's how people in computer science have been doing image processing for the past 40 years. And in the recent years, it's become clear that the same principles are used by the brain to analyze images that come into your eyes and to make sense of them. Now, how can we test that the brain is really doing what we presume it may do like an engineered system? Well, we can devise a stimulus, a picture like that. And only if the brain works in the way we assume our hypothesis is, namely that you're decomposing the image in images of different frequencies of change, high frequencies of change and smooth frequencies of change, only then will you be able to be tricked by this illusion. So if you point your mobile phone at it and ask Google to recognize what's in the picture, Google will not be fooled by it. It will be confused. If you ask an alien what he sees, he may see something else. But only if you're human, and only if your visual system works the way we presume it may work, can we trick you with this image, and only then your brain can be made to behave badly. So making your brain behave badly is for us a test of our hypothesis of how your brain may work. So since we're talking about engineering, we should look a bit about what's the hardware that we have up here. We call it wetware, because it's quite wet when you grab into it, and that of a computer, fancy computer like this one, very light, very small. So what you observe is that your cerebral cortex, that's the computational tissue that's on the surface of your brain, has something like 2.5 billion neurons. And each of these neurons, I'm going to talk about neurons shortly, has something like 10,000 synapses. So these are connections that one neuron makes with another neuron. And for example, my laptop's CPU has something like 300 or 290 million transistors. These are the smallest units of computation in a computer chip. My chip weighs 370 grams, my brain 1.3 kilogram. And we can go further. My laptop consumes 34 watts of power when this chip is on, and my brain consumes just half of that, 16 watts of power. You can't even read with the light bulb of that power in the bed if you wanted to. And then we can look at loads of other features, but what I wanted to really draw your attention to is what happens if we divide the features of one system by the features of the other system. So if we say that each synapse in your brain, so each connection between one neuron and the other one, is a computing element, because these connections can change, then we can equate that to the computing elements of a processor, namely the transistors. And you find that our brain has something like 800 times as many processors or computing elements as your big CPU in your laptop. So you have 800 times more components that compute in your brain than in your CPU of the computer. Your brain is something like 1,500 times lighter per computing element than my CPU and my laptop. But what's really the mind-blowing fact is this thing. Your brain is infinitely more sustainable. So each synapse consumes 200,000 times less energy than a transistor in my computer. Now what this means is that if we could build a computer, this chip, that is as energy efficient as my brain, or as your brain for the matter of fact, then your brain, or then that computer could run for over 400 years. So till your grandchildren on the chart that I've put on this morning. That's how energy efficient your brain operates. And this is something, one of the many things we would like to understand and learn from the brain to build better devices. So let's look quickly at what your brain is made of. I mentioned the neurons, and this is a picture, a parade example of a neuron. So neurons look something like a stellar shape, like a star a bit. They have a tree that goes up. We call that the dendritic tree. That's where all the information comes in. Then there's the cell body here, and this information is integrated. And then there are nerves that leave that neuron, nerve fibers. We call them axons, where the information flows out. And I'm zooming in now here on two regions, region A and region B, and you can here see a zoom in. And here you see loads of little, knobs, spines, as in a rose bush. And each of these spines is the site of a synapse, where one neuron meets another neuron and they talk to each other. So these spiny things, each of them is a connection to another neuron, And you can see how completely covered these neurons are with synaptic spines. And neurons are beautiful to look at. In fact, so beautiful that the gentleman who drew these pictures here of different neurons in your brain won the first Nobel Prize in neuroscience, Santiago Ramón y Cajal. He was also the guy who invented the word synapse, the word for neuron, and the whole idea that the brain is made out of a network of neurons that compute. And each of these different neurons here has in hundreds of different shapes that come, and each shape is specifically designed for a specific function. So the question is, what can you do now with these neurons? Well, we can do things that no computer can at the moment do, and it's really a challenge. So probably a few of you will have seen this picture before. In this picture, I've hidden an animal. So a few of you may have seen that. Those who have not seen it have yet a choice. You can see that animal, and I will point out where it is, but from then on, for the rest of your life, you will always see that animal. So if you don't want your brain to be spoiled, close your eyes now. Okay? So I'm going to show the animal now. It's a dog. That's the outline of the dog. And I take the outline away, so those who didn't want to spoil their brain can look at it again. Okay, so now you know that there's a dog here. And that's an amazing feat of information processing. Because somehow you make out that this is the real leg of the dog, right? This is part of his ear. This is his shoulder. There are no lines here. They're just black and white spots. But somehow you almost make out that there's an outline going here down to the foot. How does your brain do that? You can just do it naturally. You just did it in less than a second. I showed you the outline, and now you see the dog. If we can teach computers to do that, we could do a lot of things with a lot less human effort. Let me show you another example. So look at this picture here. You will see knobs basically on the structure, and you will see that some knobs may stick out, and some knobs may stick in. So let me show you this knob here. Who of you thinks that this knob sticks out? Okay. And who of you thinks that this knob sticks in? Okay. So observe this knob, and let me do something to this image. I'm just going to rotate it. So what happened to this knob? Who thinks this knob is still sticking out? Ooh, and who thinks it's sticking in? Aha. So what just happened here? This is another illustration how your brain combines information to make up perception. And the reason why you think this sticks in and this sticks out is because there's a highlight here and a shadow there. And your brain knows something about light, that most of the time the light comes from above. Right? So if light comes from above and something sticks out, you will have a highlight here and a shadow there. So your perception is in fact there to resolve ambiguity in the world. If you were just to look at this picture like that and you didn't know anything where light would be coming from, this could be as much sticking in as that one. You can't distinguish that. But you use prior knowledge about the world to resolve ambiguities in the world. So you use what you have learned to make perception happen. Okay. So let's take it one step further. So I've drawn here a set of pictures, and each of these pictures here describes an object that probably you've never seen in the real world because they were computer generated with a fractal algorithm. They're just coming out of a computer like that. And now you meet an alien, and that alien tells you this thing is a tufa. Okay? That's how they call it on their planet. These things are tufas. Now, who can point out other tufas? Do you see other tufas in this image? You can tell me the row and the column if you like. Where's another tufa? Fourth row? This one, yeah. This is a tufa, okay? Do you see other tufas? This one is a tufa? This is also a tufa? Is this a tufa? Nah. But this is a tufa, huh? Pretty amazing. So I show you one thing of an alien object, and you can tell me exactly what, oh yeah, these are tufas. This is a bit of a small tufa, but no, that can never be a tufa, I'm so sure. Right? So this is what we call one-shot learning. I give you one example, and you make up the explanation that tries to explain everything as quickly as possible. No computer can do that. You spend days, weeks, whole PhDs to teach a computer to do one thing, right? So if I tell you these two are tufas, then you're certainly more sure that this is a tufa, and there's another tufa. But what happens, for example, if I tell you this is also a tufa? Now I tell you this and these three are tufas. And now all of a sudden, your board is reshuffled and probably, wait, this is a tufa, then maybe this is a tufa. Could this be a tufa? Right? So you're reshuffling what you think and what you believe. And this is very quickly becoming very convincing. You can even have fights. I do this in my lectures, and students then argue, no, we get it wrong. It's like, I just showed you one example. So this is something we would really like to be able to understand, how your brain does that, and teach computers to do that. Oops. Okay. So what we really do in our lab is what I call reverse engineering the brain. Devising experiments where we make your brain behave badly so we can test what your brain is doing, and then use that to understand how the brain goes from perception, like seeing where this ball arrives, how it does the information processing in neurons, in synapses, does the learning, and how it then controls the movement of muscles to catch this ball. And we really think you have to look at this thing in a loop, because you constantly perceive and you constantly act, and you cannot separate perception from action. And action does not come before perception, just as much as perception does not come before action. It's a constant loop. And this means you have to think about how the brain can resolve these things in this manner. So let me show you something about your brain. So here we show a classic experiment. This experiment was an experiment that won the gentleman a Nobel Prize in the 1960s. So basically what they did is they showed on a computer screen a white dot that moved. And that dot, as it moves across the retina of the eye of a subject. And then you can look at a zoom up of that, and there you can see an individual neuron. That's the white dot that comes here through the lens into the eye. And whenever this white dot moves over the dendritic tree of these neurons, you will see that this neuron is light sensitive and starts to fire what we call action potentials, or spikes. And I'm going to make you hear them. Let's see if this works. Right? So this is this little area where the neuron is most sensitive to, and as the experimenter moves the white dot through, you can hear the firing of individual brain cells. Different shapes to find out how big is this, what we call the receptive field of the neuron, so the region where a neuron is sensitive to. So they use this ring and try to cover up some parts, see whether the neuron still responds. And with these type of experiments, we were able to characterize, or we, as my colleagues in the past, were able to characterize the properties of individual neurons. And we know how these individual neurons work very well. If you just see a small example of the world, you're just limited in what we can say about the world. And this is one of the most important reasons why you can't just understand what the brain does by looking at single neurons, from simple examples like that. So therefore, we have to think about what do circuits in the brain do. And here we have a fascinating new technique. It's a genetic technique that enables us to stain each single neuron, each single brain cell in a different color. That's because each cell decides to color itself in a random color of its choosing. And the most important thing I want you to notice is not the colorful bodies of the cell. So this is the cell body of the cell. This is one cell, and these are loads of different cells. I want you to look at this felt work. This looks really like felt. But this is not felt. This is these dendrites and axons. They're so densely bunched up in your brain that there's virtually no free space. In fact, you get, of these little guys here, you get 4 kilometers per cubic millimeter of your brain. 4 kilometers per a volume that's the volume under a well-trimmed fingernail. 4 kilometers of that stuff. That's how densely your brain is connected. And it's this connectivity power that makes us special. So we're not the species on the planet that has the most neurons. We're not the species on the planet that has the biggest or the smallest neurons. We're the species on the planet that makes the most connection between the neurons. So once we have this fantastic brain with all what it does, we would like to be able to help people, for example, who have lost the ability to move their arms because they've lost their whole arm, because they had a stroke, they're paralyzed, or had a spinal cord injury. So this thing, what we call here, is a brain-machine interface. It's an interface that tries to directly read out from the electrical activity of your brain what the animal, or the human, in this case it's an animal, what you want to do, so how you want to move an arm. And this is an experiment that we've done. I'll just show you what's going to happen here. And so this is a robotic arm controlled by a chip implanted in the brain of this monkey. He's giving food, and he can feed himself with that. Okay. So this monkey has his arm, it's just tied to his back, so he didn't lose the arm, but the robotic arm here is controlled from the brain. Now the problem is, what you don't see here is that actually we don't know what's going on inside this brain. Instead, what my colleagues did is they recorded loads and loads of sessions where the animal was moving his arm in a natural fashion, and then correlated that with activity inside his head, and then tried to work out if the activity looks like that, then the arm looks like that. So we don't really understand what the monkey is thinking here. We just see that the pattern of activity looks like a movement of the arm. Okay? But this is what we call brain-machine interfacing, really trying to link up what's going on inside you with the outside world without you having to type on a keyboard or use a computer mouse. And this brings us to a very simple question. Namely, why do we have brains in the first place? Why do you think we have brains?
Central control.
Central control. Other ideas?
Predict danger.
Huh?
Predict danger.
Predict danger. For movement. That's a student of mine. Any other suggestion? Processing and store information. Okay, so let's see. All these guys here have brains, and all these guys here do not have brains. Right? A mushroom, a tree, a flower, a coral don't have brains. Here you have an octopus, a person, a mouse, a fly. They do have brains. What do these guys also not do that these guys do? Move. So somebody could say that maybe the only reason why we have brains is because we can move. Because if you can move, then you can do something about if danger comes along, because then you can try to predict the danger and step away. You may need to remember where are good places to step into, for example, to forage for food, because you can move there. So really the idea is that, in fact, that's a fact, there's no organism on the planet that doesn't move that doesn't have a brain. Okay? All organisms that move have a brain. And there's a classic example. It's a guy that swims here around the sea. It's called a sea squirt. And that sea squirt is an animal, it moves, and then it sucks onto a coral, and the first thing that it does when it's sucked onto a coral is that it starts digesting its own brain. Yeah? And that's what some of my colleagues do when they get tenure. But if you don't move anymore, you don't need a brain. Okay? So this is not textbook knowledge. If you write this in an exam outside of this university, you'll probably have trouble with your teachers. But I really believe in that. The only reason why we evolve brains is because we have movement. Okay, so something about movement and central control came up. So this question here becomes important because when we try to build artificial systems that we want to control and move, it's actually really, really hard. So this is a PhD thesis that was done in a lab in Germany. This young man, bright man, spent 3 years of life doing programming or teaching this robot how to stack cups. And this is the video, this outcome. Robot can stack cups and push them away. After 3 years of work, that was pretty impressive. But sometimes, no, this was really an impressive piece of work from an engineering perspective. If the cups are not exactly placed in the right place before it starts, the robot fails. So now let's look at another younger gentleman. He doesn't have a PhD yet. He doesn't even have his primary school completed. This is the reigning world champion in cap stacking. And then he thought, oh, he wasn't sure whether the start was right or wrong, and he does it again with the ease of an 8-year-old boy. Okay? So this is really where you like to understand, how the hell can we do that? What is going on inside our brain that allows us to do that and not do PhDs about simple cup stacking for 3 years? Oops. No, I'll just do that. Okay, so what I'm going to talk a little bit more now is try to understand how the brain controls movement and learning. And that's important because we want to know how the brain works. I do. We want to help people who have movement disorders whose ability to move has decreased with aging, whether they have some neurodegenerative disease, for example, Parkinson's or multiple sclerosis, or just simply to be able to build interfaces that allow brains to more directly connect with machines. And the applications are prosthetic hands, general robotics, help people in rehabilitation, or build simply better and nicer computer user interfaces. So let's stick to the hands for a moment. So here you see 3 versions of current hands. So this is Jamie Oliver's hand, very skillful, separating the yolk from the white. Here you see the Otto Bock prosthetic hand. So that's a standard prescription prosthetic hand that's given out to patients at the moment. And this is one of the most sophisticated, or probably the most sophisticated robotic hand, the Shadow Robot hand. Okay? So the amazing thing about this, he just does it. This can't do what he does. This could do what he does, i.e. he, Jamie Oliver, but we don't know how to move these individual joints. I know that if I press a button, then this joint will move, but I don't know how to coordinate these joints to make something like separating egg from yolk. And so we basically started with a very simple question a few years ago. What are the most important hand movements? You can make a quiz show out of it. I guess your job by, or your profession by showing me your characteristic hand movement. And if you look into the literature, the textbook says, well, the most important hand movement that a human makes is the power grasp and the precision grip. Fundamental. You can't do anything without it. Any doctor that puts you in a rehabilitation, for example, after a stroke or an injury, will make you rehabilitate on the power grasp and the precision grips. So we asked, okay, is this actually true? Because if you look at the literature, you go back to 1810, when some doctor, GP somewhere in Sussex said, these are the 2 most important things. And since then, generations of academics and doctors have cited this gentleman and went past and long forgotten what the evidence was that these 2 are the most important hand. So what we did is we had one of these fancy so-called cider gloves. So that's a glove that you wear on top of your hand, and each joint of this glove can measure the movement of each individual joint. And here you see my grad student Anastasia, and so we did experiments where people were wearing this glove for a whole day, from the start to the end. You go to breakfast, you go to the bathroom, you work at the desk, whatever you do, you wear this glove, and let's just collect data. And what we found after all that time was you didn't do a single precision grip. You may have done a bunch of power grasps, but this almost never occurred during the whole day. So our whole understanding, we then realized, about what are the most important movements is completely anecdotal. It's actually not even scientific, right? It's less than stamp collecting or drawing pictures of birds. So what we then set out is to try to understand what are the real measures, what are the most important movements, and then basically we used so-called machine learning algorithms that are computer algorithms that basically try to find from data, and just from data alone, meaning inside that data without making any assumptions about what this data may be about. Right? What are the structures that tell us something about the data itself? Keyword here, you might have heard of this idea of big data analysis. Okay, so we did that and we found that the 3 most important movements were something that looked like this, something that looked like this, and something that looked like that. And if you take these 3 movements together, you can compose 90% of all the movements that you make during a day. So these are a bit like the letters in an alphabet, or so to speak, the prime numbers of hand movements. So as already mentioned, what we're talking about here is really to go from the genome, where people studied and analyzed individual genes by looking at the base pairs in the DNA, to the proteome, which was in the structure of proteins, to what's now going on, the connectome, so knowing which brain cell is connected to which brain cell, to what we call the ethome, from the Greek word for behavior, to characterize what are our natural movements actually. And why is that important? Well, let me first show you how we did it. We did that by basically developing solutions where we have sensor suits that can measure the movements of the joints of the body, as well as this data gloves that we found, which you can wear in your hands, and allows to collect data in daily living situations. So not an artificial lab situation, but your natural behavior, how you would behave when you are in the bedroom, when you make yourself breakfast, you eat and clean up, or how you work in the office. So this was a real simulations of daily activity life, and we built the world's first so-called atomic database. And this just shows you a snapshot from the database. So here you see the joint angles of the individual, the arms, the forearms, and the hand, how they move. That's the right arm, that's the left arm, that's a snapshot of what the leg does. So this is all squiggly data like that. And then we knew what the person was doing. He walked, then he sat down, then he picks up something from the floor, and so forth. Right? So then we can try to understand how do these activities here relate to these movements in a natural setting. And what we can do with that is now the true power of this approach. So at the moment, we're trying to use to detect subtle changes in your daily movement behavior to detect early onset of Parkinson's, early onset of multiple sclerosis, and early onset of Alzheimer's. Think about it that way. Most of the time when people have neurological disorders, aside from the fact of headaches, they will notice in their partner or in themselves a subtle change in behavior that becomes then so persistent that then the patient has to make up his mind, go to a neurologist. That neurologist has to see him for at most, 20 minutes? And then from that pattern speaks, okay, sir, your hand's shaking at a characteristic 7 hertz. You may have developed a low form of Parkinson's now. Now that's one way of doing that. Anecdotal, somebody observes a subtle change, a doctor sees you for 20 or just 10 minutes and tries to see from your changes in behavior pattern what's going on. Or we can have a computer system track your movements with simple sensors that you can have sewed into your clothes that will detect changes in your behavior patterns. We want to do that for sleep deprivation, so we want to know, for example, a driver is falling asleep because his movements are slowing down and waking up or preventing from starting a car. We can detect depression with that. That's fairly easy. You can do that. But I'm not in a good state now, but really found seeing this state here. We can read that from the normal body language of the person. And of course, questions about deception. But if we know how people normally behave and move and coordinate their joints, we can also use that to build smarter prosthetic hands, using, for example, the smart hand movements I showed you. We can build better robotics that we can use in factories to make precision tools. And we can build better assistive devices that help people who are disabled move in the environment again. Okay. So what we've done so far, as I already mentioned, is we built this database of behavioral biomarkers for diseases. We applied that. We built a large database, and these are my collaborators, a large database of 44 subjects of healthy behavior. We've screened well over 127 patients actually now, and we can now, with 92% detection, say whether you're developing arthritis of the knee joint based upon subtle changes in how you walk. And as I said, we're just currently screening that for further diseases. Okay. So I want to take you now a step further, and I'm going to jump over a few things because I want to tell you something that I find important. So having spoken about these diseases, we can ask ourselves, what about once you have this disease, so we're not able to detect it and do something about it? Let's assume you suffer from Parkinson's, multiple sclerosis, a form of muscular dystrophy. You had a spinal cord injury, a stroke, or an amputation of your upper arms, or simply a degradation of your musculoskeletal system due to aging. In all these cases, your movements of your arms may and hands will or may be impaired progressively. Now, one thing that happens, though, independent of any of these conditions you suffer, you retain one ability to move, and that's the ability to move your eyes. And this is retained because your eyes are directly connected to your brain. They don't go via the spinal cord or the brainstem. In most of these disorders, spare the eyes movements. So to the very late stages of the disease, you can virtually always move your eyes freely. So here you can see how we basically draw the rays shooting out of the eyes, showing that the eyes can track objects. This is an early experiment we did where we just simply looked at how people move their eyes to look at the scene of breakfast. So this is how you look in the world, you jump from one place to the next and focus from one place to the next. So this technology, these experiments are well known, they've been done for over 50 years, and they typically use technology that's very expensive, so-called eye trackers, that cost around 20 to 30,000 pounds if you want to have a proper system that measures things accurately. Instead, what we did, we went out and basically hacked a few video game equipments that we could find lying around, and basically developed a system that didn't cost 20,000 pounds, but just 10 pounds to make, that has the same accuracy and performance as the conventional system. And we did this a few years back. So now we were able to measure the movement of the eyes, be it in patients or in healthy people, but in both cases they always have this ability to move their eyes freely. And the idea was then that once you know where the pupil is looking, you can then work out, if these are the two eyes, where the person is looking. And you can then use that to control, for example, a mouse pointer. So as you look over a computer screen, the mouse pointer follows where you look. And we demonstrate that here. Oops, this is a video which should play by playing a video game, namely the video game Pong. So you see here a patient lying in his bed, and as he moves his eye, we can, for example, allow him to control this paddle that follows the movements of his eyes like a mouse pointer would do, and this paralyzed patient can then play an interactive video game. And this is the first demonstration that you can use a brain-machine interface that exploits the movement of the eyes to control, in real time, very, very fast, action-paced video games. And you can do more than that. You can, for example, browse on a computer, because if your mouse pointer follows your eyes, then you can just, for example, wink to issue a click. And then you can navigate the web, or we can display a keyboard on your screen, and you can basically click your emails together by looking at these things and clicking. It's a very simple solution, and we wink because, of course, you naturally blink, so we don't want to execute a command only because you blinked. But wink is something artificial that you can retain control over. So this simple interface allows us to build what we call a task-level brain-machine interface. There's no need to go into the details of this picture. All I want you to show is the two axes here. One, that's cost for a device, inclusively training or operating on patients, and this axis goes up by a factor of 10. So this is $100, $1,000, $10,000, $100,000, $1 million. And this is something a bit more abstract. This is what we call the readout performance. So how much information we can get out of a person to control a device, like a mouse pointer. And this monkey interface that we showed you at the beginning, that's devised to have that plus the operations and the implants for a patient cost currently around $400,000 to $500,000. And you need to train, so the current patient trials that they're running, this is done in the US, patients need to train for about 6 months to be able to click yes or no. So a binary choice. Then you may have seen loads of these things where people have like caps where the electrodes reading of the brain, you don't need vibration, it just sits on your brain. These devices cost around 30,000 pounds, and it takes you quite some time to learn that, so you have a few weeks to learn. Well, this device here, our device here, costs around, as I said, 10 pounds to manufacture, and you can use it and learn to use it in less than 5 minutes. And in fact, I can show that to you because, you can go outside this exhibition at the end of my talk, and you will see a booth that looks like that. And that's our eye tracking device, and you can yourself play this game Pong. And so far, we did this as part of 2 exhibitions, the Strictly Science exhibition that we organized a few weeks ago, and it's part of the Imperial Festival. We had over 2,300 people using this device, and within 5 minutes they were able to beat a human, fully body-able computer player who was using a gamepad at playing this game. More than half of that. So a training time of less than 5 minutes, and an ability to read out information from the brain that is, in terms of its speed, so to make reaction possible beyond any other device currently on the market or in research. Okay. So, of course, the media saw this as a feast because, oh, video game playing with devices. And I was a bit embarrassed by that at the beginning. So like, my God, I'm doing here serious research and they're going all about the video gaming. But what actually then touched me is that we got a lot of letters. Letters from patients, letters from parents, letters from children whose parents were paralyzed, who were saying, this is fantastic. I've never been able to play a game with my father. I've never been able to play a game with my son. And this device would enable that. So that alone gave us actually a boost in motivation that we hadn't done something completely useless. But what we actually want to do is to be able to do that. Use the same interface here to allow a prosthetic arm, here just a commercial robotic arm, but later it can be prosthetic arm, to allow you to drink a glass of wine without the assistance of a carer. But the whole user interface just still costing you £10. Okay. And with that, I'm coming to the end of my talk. So I've shown you work that I've done myself, my PhD student William, my PhD student Andreas, Konstantinos showed, and Margarita did the work on this atomics data, and I hope you enjoyed it, and I'm open for questions.